Welcome. Today, we are focusing on one of the most pharmacologically significant families of proteins in the human body, the G-protein-coupled receptors, or GPCRs. GPCRs represent the largest and most diverse group of membrane receptors in eukaryotes. More importantly, from a clinical and pharmacological perspective, they are the target for approximately 30% to 40% of all FDA-approved drugs on the market today. From managing hypertension and asthma to treating psychiatric disorders, manipulating GPCR pathways is foundational to modern therapeutics. Moving on with the structural overview, GPCRs are often referred to as 7 transmembrane receptors. They consist of a single polypeptide chain, exactly 7 times. Now let's examine the extracellular domain. It contains the N-terminus and various loops. This is the primary binding site for hydrophilic ligands like peptide hormones or neurotransmitters. We will now consider the transmembrane domain. The 7 alpha helices form a structural core. Small hydrophobic ligands often bind deep within this core. Finally, let's examine the intracellular domain. It contains the C-terminus and intracellular loops. This region interacts directly with the heterotrimeric G proteins inside the cell. Let's discuss the mechanism of action. A GPCR relies on its coupling to a heterotrimeric G protein, consisting of 3 distinct subunits, alpha, beta, and gamma. In the inactive state, the receptor is unbound, and the G protein alpha subunit is bound to GDP. An agonist binds to the receptor. Agonists can be neurotransmitters, hormones, or drugs. This induces a conformational change in the structure. The structural shift prompts the alpha subunit to release GDP and bind a molecule of GTP. GTP is taken up from the surrounding cytosol. Binding of GTP activates the G protein. To release GD, the alpha subunit detaches from the receptor. It dissociates from the beta-gamma dimer. Now both active components can regulate downstream effectors. The alpha subunit interacts with specific enzymes like adenylyl cyclase to generate second messengers. The signal is self-terminating. The alpha subunit possesses intrinsic GTPase activity, eventually hydrolyzing GTP back to GDP. The subunits reassemble. That concludes our overview. The alpha subunit possesses intrinsic GTPase activity,